Genetic engineering. Genetic engineering is the direct manipulation of one or more genes. In short, genetic engineering is the process of taking genes from one organism and adding them to another organism in order to give the second organism a desired trait. Create new crops that are resistant to pests and diseases. Develop new medicines and vaccines. Treat and prevent genetic diseases. Improve the quality of life for animals. However, genetic engineering also has the potential to be harmful. For example, it could be used to create organisms that are resistant to antibiotics, or to create organisms that are harmful to the environment. Specific applications of genetic engineering. Agriculture. Genetic engineering has been used to create crops that are resistant to pests and diseases, as well as crops that are more nutritious. Medicine. Genetic engineering has been used to develop new medicines and vaccines. Animals. Genetic engineering has been used to improve the health and welfare of animals. It is important to carefully consider the risks and benefits of genetic engineering before using it. In-depth overview of genetic engineering. Basic principles of genetic engineering. DNA. Genetic engineering involves working with the genetic material of an organism, which is composed of deoxyribonucleic acid, DNA. Recombinant DNA technology. Genetic engineers use recombinant DNA technology to introduce, remove, or modify specific genes within an organism's DNA. Genetic tools. Techniques such as gene cloning, polymerase chain reaction, PCR, gene editing, e.g., CRISPR-Cas9, and gene synthesis are commonly employed in genetic engineering to manipulate DNA. Techniques and tools in genetic engineering. Gene cloning. Gene cloning involves isolating a specific gene or DNA fragment and producing multiple copies of it using recombinant DNA technology. Polymerase chain reaction, PCR. PCR is a technique used to amplify a specific DNA sequence in a test tube, generating millions of copies of the target DNA. Gene editing. Gene editing techniques, such as CRISPR-Cas9, enable precise modifications of DNA sequences within an organism's genome. Synthetic biology. Synthetic biology involves designing and constructing new biological parts, devices, and systems by combining genetic components. Ethical considerations and challenges. Controversies. Genetic engineering raises ethical and societal concerns, such as the potential for unintended environmental impacts, the safety of genetically modified organisms, GMOs. Regulatory frameworks. Many countries have established regulatory frameworks to assess and manage the risks associated with genetic engineering. Public perception. Public opinion about genetic engineering varies widely, with concerns about potential risks, perceived benefits, and the need for transparency and public engagement in decision-making processes. Importance of genetic engineering. Advancing agriculture and food security. Genetic engineering enables the development of genetically modified crops with improved traits, such as increased yield, enhanced nutritional content, and resistance to pests, diseases, and environmental stresses. Disease prevention and treatment. Genetic engineering plays a crucial role in the development of therapies and treatments for genetic disorders. It enables gene therapy, which aims to correct or replace faulty genes responsible for genetic diseases. Environmental conservation and sustainable practices. Genetic engineering can contribute to environmental conservation by developing genetically Modified organisms, GMOs, with improved abilities to clean up pollutants, degrade waste, and promote sustainable agricultural practices. Scientific research and understanding of life. Genetic engineering provides valuable tools and techniques for scientific research, allowing scientists to study gene functions, regulatory networks, and fundamental biological processes. Industrial applications and biotechnology. Scope of genetic engineering. Agriculture and crop improvement. Development of genetically modified, GM, crops with improved traits such as pest resistance, disease resistance, herbicide tolerance, increased yield, and enhanced nutritional content. Improvement of post-harvest traits like shelf life and nutritional quality. Medicine and healthcare. Gene therapy. Treating genetic disorders by delivering functional genes to replace or repair faulty genes in human cells. Production of therapeutic proteins and antibodies using recombinant DNA technology, leading to advancements in the treatment of diseases. Personalized medicine. Tailoring medical treatments based on an individual's genetic profile. Biopharmaceuticals and industrial applications. Production of valuable pharmaceuticals, enzymes, and other biochemicals using genetically engineered microorganisms, plants, or animals. Environmental conservation and bioremediation. Creation of genetically modified organisms, GMOs, with enhanced abilities to degrade pollutants. It's important to note that the scope of genetic engineering is constantly expanding as new discoveries, technologies, and applications emerge. With ongoing advancements in gene editing tools, DNA synthesis, and our understanding of genetic systems, the potential for genetic engineering to address various challenges and benefit society continues to grow. Ethical considerations, regulatory frameworks, and public engagement play crucial roles in guiding the responsible and safe application of genetic engineering.